This video is part 2 of my presentation of membrane transport. On the first video, I was talking to you about the mechanisms of passive transport, including just regular diffusion or plain diffusion. I also talked about osmosis and facilitated diffusion, which requires the participation of transport proteins. Now I'm moving on to the next two categories of membrane transport, which are going to be active transport and bulk transport. Bulk meaning moving materials in large amounts. But first, active transport. And so in active transport, we're going to see two characteristics that are the opposite of passive. In active transport, particles are going to be moving against a concentration gradient. Let me see if I can have the setting to, yes, work as a pen. So here we go. So particles are going to be moving against a concentration gradient. And what this means is that particles are going to be moving from low concentration to high concentration. Previously, I have used the analogy of a person on a bicycle at the top of a hill and just allowing gravity to take them down to the bottom of a hill. Now imagine that the same person on a bicycle wants to go back to the top of the hill. Now they have to go up against the slope from low to a higher elevation. And that is going to require a lot of energy for pedaling, for making that bicycle move. Likewise, in a cell, when the cell wants to move particles from lower concentration to higher concentration to a place where particles are already heavily packed, the work can be done, but this is where the second condition comes into place. For active transport, the cell will have to pay a price and is going to have to use energy in the form of ATP. So this is how active transport is the opposite of passive. In passive, high to low, no energy. In active, from low concentration to high concentration, but yes, the cell has to be spending energy now. And one of those examples that I have mentioned previously about active transport includes the sodium-potassium pump, which allows nerve cells to create an electric potential. And this illustration really shows how this sodium-potassium pump works. A first solute can be something like sodium that is going to be going out. This is going to be, for example, a, let's see if we can write, sodium ion that is going to be moved from where it's low concentrated to where it's more concentrated. And potassium will be going in the other direction, but it just follows sodium at first. This sodium is going to bind to a region of the transport protein where it can fit. Next, ATP does what it knows how to do best: phosphorylate, give up one of its phosphate groups, and in doing so, it's transferring a little bit of energy that is going to be used for changing the shape of this protein. And now look at how this protein is opening to the opposite side from what it was first open. Then, maybe I should be using my laser pointer. Yes. So here we go, sodium moving into this space in the transport protein. ATP allows this protein to change its shape, opening to the other side, and then releasing the sodium ion. This protein can be used to do another work while it's still energized, while it's still phosphorylated. And it can pick up a second solute, and this second solute can be something like potassium. And this potassium ion is also going to find a place where it can conveniently fit into this transport protein. As the protein releases the phosphate, it returns back to the original shape. This shape over here is the same one you see down here, dumping this potassium in a place where it's more concentrated. So potassium goes from where it's less concentrated to where it's more concentrated, and sodium did the same thing from low concentration to high concentration, always against the concentration gradient, from low to high concentration. And the condition of energy was fulfilled when ATP was used to change the shape of this transport protein to allow it to do the work. So that's active transport. And sometimes cells need to do that, like in the case of nerve cells. And the last category for membrane transport is going to be known as bulk transport. This is when materials can be moved in large amounts. If the movement is to the outside of the cell, like you see here, this is a transport vesicle. One can guess maybe this transport vesicle came from the Golgi apparatus. And these are proteins or some other material the cell wants to export to another part of the body. So the transport vesicle is going to be fusing with the plasma membrane. Notice that the vesicle has the same primary structure. It's a phospholipid bilayer, and so it can easily merge with the plasma membrane. When the transport vesicle fuses with the plasma membrane, it will be opening to the outside, dumping out at once all of the contents. All of these molecules are being released in one single step, rather than moving them one at a time through transport proteins in the plasma membrane. So sometimes cells want to do this exporting, this releasing of materials in a fast, expedited fashion. This is, for example, how insulin is released from your pancreas. When you eat something sweet, you want your cells to quickly take that sugar if they are in need of it. And so insulin will be released from your pancreas cells by the process of exocytosis. Just to give you an example. The opposite can also be done. Sometimes cells can form a pocket with their plasma membrane. This pocket can collect a droplet of fluid with many of those particles in solution that the cell wants to take. And so when this pocket forms, it eventually ends up closing it around, forming a vesicle, pinching off, and traveling to the inside of the cell. If the material being brought inside is like a droplet of liquid, this is going to be known as pinocytosis. If, however, the cell happens to engulf a larger item, not just particles or molecules, but like entire cells. Here you see, for example, an amoeba, and this is a single cell protist, and it likes to feed on other types of single cell organisms, like a group of algae over here. There's a cluster of algae. They've been surrounded by the amoeba as it closes in around, using its own plasma membrane to form a food vacuole that will later digest those cells. When the cell is consuming a bulkier item like another cell, this is going to be known as cell eating or phagocytosis. And there's another type of endocytosis, which is known as receptor mediated endocytosis, and this is going to be more specific. Now the cell doesn't want to just take everything that is on the outside. The cell may be interested in only taking certain specific types of molecules, and what it will have to use is specific receptor proteins on the plasma membrane that can bind to the ligand. This is going to be a material that the cell will want to import, and then when the material has been collected, special coat proteins are going to be help pull in the plasma membrane to form a pocket, to form a vesicle. But the key here is the difference compared to the previous just pinocytosis is that the cell can bring in specific types of molecules it wants from the outside, not just everything, not just anything that is on the outside of the cell. So from the outside to the inside, this is called endocytosis, and this specific case is called receptor-mediated endocytosis. There's going to be a video that is produced by our textbook on membrane transport that is going to be showing animations of how this works, and I want you to watch that as well so that the two-part videos that I have produced will make more sense to you. That is all for now. Happy studying.